but also justify why we're using the sheep brain. Sheep brain is cheap, but the most important thing is that it's very similar to a real human brain that I brought here. So it's always useful to do kind of the direct comparison. So I'm gonna use this. And so what are the differences here between this adult human brain fixed in same chemicals and the sheep brain?
Let's talk about size first.
Okay, size. What else?
They have very similar, looks like overall structure with the ridges and the stem.
Yes.
The gyri and sulci.
Right, exactly. And notice anything similar or different here if you go to the bottom?
Oh, the cerebellum is relatively larger on the human brain.
On the human brain, yeah, it's pretty big. It's it's pretty big relative in the sheep brain as well.
Yeah.
But anything else?
The olfactory bulb
Yes.
is prominent on the sheep,
Yes.
not so much on the human.
Very prominent on the sheep, exactly. And they're just small vestiges on the human brain. So let's go back to the ridges. So all of this infolding, what is that good for? All the infolding that you see on this outer covering or cortex. Why is that important?
Yes, you were talking about surface area.
And what about surface area? What is good or useful about that?
It increases it so you can jam more in a little space.
Right. So it increases basically the computing power of all of these cells that are making up this infolded structure is helping the computing power of the brain. We have a lot of computing power. And that actually means that the sheep might have a surprising amount of computing power. What do you think those sheep think about all day?
Food.
Food? Okay.
Survival.
Yeah. Anything else?
Mating.
Mating. But is that different from other? Oh gosh, I'm getting a whiff of formaldehyde. Actually, what we know is that the sheep visual system is very, very advanced, such that individual sheep can recognize all other sheep in their flock and discriminate them from other sheep that are not in their flock. It's actually quite a developed system, and they do tests on visual acuity in the sheep. So that's one thing that the sheep seems to be even better than most organisms that need food and need to reproduce. Their visual cortices are particularly strong, and that's back here. And then you can follow it up. The olfactory nerve is right here. Here's the optic chiasm, that crossing. So your eyes and the human are sitting right here, and all of the information from the right visual field goes to the left side of the brain, and all the stuff from the right visual field goes to the left part of the brain. And let's see. This is the vestige of that cross right there. Let's see if we can see. This is better. You see that cross? That's the same optic chiasm. So it's, sorry. The other, besides general organization, the other things that are really, really similar that's fun to go over in class are the cranial nerves. So there are 12 sets of cranial nerves. And the cranial nerves differ from your regular spinal nerves because they're inserting in the brainstem or midbrain, or it goes directly into the cortex. So they're specialized nerves, starting with olfactory. They cover the special senses, head and neck, sensory and motor systems, but you can follow them down. And 1, 2, 3 are always easy to see, and then it gets a little bit harder. So 1 is olfactory. This is kind of worn out here. 2 is the optic chiasm. And 3 is this flap right there. That's a nerve. And let's see if we can find it here. This might have been kind of torn off. The other thing that you can see really well, if you come and put your face over here, if you look down there, can you see 2 little bumps?
Yeah.
Yeah?
Is that what, mammillary body?
Yeah, mammillary bodies. So looks like a couple of little mammillary glands. There's those two little bumps right at the bottom there. And can you see it here? They tend to get squashed. Oh, but look, there they are. Those two little balls right there are the same mammillary glands. Okay, so let's move on to the basic lobes of the brain. So frontal lobe, same as sheep brain, right at the front of the brain. Your eyeballs are right here. The sheep eyeballs are kind of more off to the side here. Big lobe back here, occipital lobe, important for primary visual senses. The lobe between the frontal and the occipital lobe is right about here. That's parietal lobe, and that would be located right around here. And the last lobe is this lobe right here, the temporal lobe. And you can see it not quite as well differentiated, but it's this region right here in the sheep brain. And that houses two of your favorite structures that I heard you talk about, the hippocampus and the amygdala. What was your favorite structure?
What did I pick? I picked the whole temporal lobe.
Oh, well, see, you're all temporal lobe centric. It happens that the hippocampus is my favorite area as well, so I've studied it for the last 25 years. And it is deep in the temporal lobe right here, kind of below. It's a tube-shaped structure in the human that goes right here. And one of the things I want to do with some of these extra brains is to do a hippocampal dissection for you for the sheep brain, because it's really fun to pull it out and see. The difference between, one of the main differences between the human brain and the sheep brain is the configuration of the hippocampus. So in the human, it lays flat in the deep part of the temporal lobe right here. And in the sheep brain, it starts down here, but it actually, starting down here, it curves up. And so the top part is way up here, and the bottom part is down here. So we're going to see this C-shaped structure once we go into it. Even though it's a different configuration, it has very similar functions. So as you mentioned, memory, spatial orientation, absolutely, that's exactly what it's doing in both the sheep brain and the human brain. Okay, so those are the lobes. Oh, and then, sorry, let me just go through a few more of these. So this is the pons. The pons is the structure right opposite to the cerebellum. It's a lot of the fibers from the cerebellum are going through the pons. And down here is the medulla oblongata, where a lot of the cranial nerves have their origin cells are in the medulla oblongata. Okay, so now I want to have you guys why don't you start with some photos of just the overall structure to illustrate what we've talked about so far. So where are the lobes? Top view, bottom view. So your atlas can start with that. And then we'll start kind of cutting into it. That can and should be labeled on these kind of overall views of the sheep brain. Obviously the cortex, information about the folding, what that means. What are the major functions of these major lobes that we talked about? What does the frontal lobe do generally?
That's the
Cognitive.
Can you
control.
Motor
Okay.
commands.
Motor commands are a little bit farther back. Personality. If you damage the prefrontal lobotomy that they did in the 1940s, basically separating out the cutting the connections between the frontal lobe and the rest of the brain, it changes your personality. Working memory is dependent on the prefrontal cortex, that keeping in mind memory. It's different from the memory dependent on the hippocampus. It's the memory that allows you, I always used to say, I give the example of keeping a phone number in mind between the time you look it up and the time you need it. Of course, you don't need to do that. I have to think of another. What's another memory example?
Remembering couple stuff for shopping.
Exactly. Yeah. The list while you're shopping, or even remembering somebody's name. Somebody just tells you your name, and it's that working memory that, for me, often fails, and I can't remember their name anymore. Okay, so that's the frontal lobe. Occipital lobe, we already talked about. Primary visual cortex. What about parietal lobe? What is that? What is that area involved in?
Sensory perception, like how are you holding yourself and everything like that.
Mm-hmm. So,
proprioception,
Proprioception.
The body senses, basically. Right?
So one of the newest and exciting areas that have focused on the parietal lobe is decision making. It's not just a motor area, but it's really important to help you make cognitive decisions. It's also an area that, the example that my students, the undergrad students, love is a lesion in the right parietal lobe. What would that do to you? And
oh yeah, is that the hemi neglect?
yes,
Syndrome?
hemi-neglect, yeah. So
what
right,
Hemi.
neglect. So right parietal lobe lesion is processing information from the left side of your space. And so what happens is you have a lesion on the right side, and I ask you, please draw a clock, okay? You'll draw a clock, but you'll only put the numbers on the right side, not on the left side. You will have a plate of food in front of you, and you'll only eat the food on the right side of the plate because you're basically ignoring the left side of the plate. So it's attention, spatial attention, decision making, and some motor functions as well. So it's a very, very complicated area. And then finally, we're all experts now, or you guys are real experts on the temporal lobe. Higher order visual processing, long-term learning and memory through the hippocampus, and emotional processing via the amygdala, all down there in the temporal lobe. So those things can be pointed out in the pictures once you make the atlas. What about the cerebellum? What does the cerebellum do? Balance, yeah, exactly. So if you have damage to the cerebellum, even something as I was going to say simple. Walking is not that simple. It's actually a very, very difficult, coordinated process that becomes much more herky-jerky, and you can't attain the smooth movement that the rest of us have. All right. So why don't we move on? Oh, sorry. And the last thing I wanted to point out that I'm sure you're all aware of is the overall structure. All brains have a right hemisphere and a left hemisphere. Everything is doubled. There is hippocampus on the right, a hippocampus on the left, a thalamus on the right, a thalamus on the left. So why would that be? You kind of take it for granted. There's this kind of separation here. What purpose do you think that might serve?
Well, we talked about the
Redundancy.
right taking care of the left and left taking care of the right. So I guess you still have higher chance of survival if one of them goes wrong. People have lived with.
Yes.
Half
Yeah, absolutely. Yeah. There's redundancy. That's good.
Yeah,
Any,
what she said.
good. Excellent answer.
Humans are symmetrical beings.
Yes, there's symmetry in our whole body.
Everything, yeah.
So it makes sense that the brain also follows that symmetry. The truth is, if you go to the textbooks, they pose that question, but they don't answer really well. I mean, we don't know. It's part of our evolutionary organization that there is symmetry in all of our body and then all of our brain.
How far back down the evolutionary tree do you have to go to get to non-symmetrical brains?
Yeah, there are, even in the neural nets, that's a really good question. I don't think when you go down into worms or insects, insects have symmetry too, right? There's the right claw and the left claw. but they don't have nearly as clearly demarcated symmetry in the brain. They tend to be organized with neural nets rather than, you know, this very complicated structure. But as long as you have symmetry, even a worm, there's a right side of your body and a left side of your body, but that is completely controlled by nets that don't have a clear right-left symmetry. So that being said, it goes back a long way. So nature has shown that that's a very valuable thing to have. And for dissections, it's also great because it gives us a perfect first way to cut the brain. So why don't you take your knives and try in a nice smooth cut to go from the front all the way to the back, and we'll take our first look inside.
Okay. Watch your fingers.
Yeah. Try not to saw. So my recommendation is to like that.
Like birthday cake.
Like birthday cake, exactly.
Okay, we're now starting the first cut. And this patient has been anesthetized, so don't worry, students. He's not feeling any pain. Nice.
All the way through the stem.
All the way through the stem.
Please don't.
Nice. Okay, great. Perfect. Yeah, go ahead and take some pictures of that first cut, and then we'll go through some of the major structures that you can see in the middle of the brain.
Okay.
It really gives you, you can take your gloves off and
Yeah.
get new ones, yeah.
So do you usually teach your students first the anatomy, or you just have them do an experience like this? What do you find most helpful?
You know, in my brain and behavior class, I give them an overall lecture with pictures, so they get the kind of just overall view of what to expect, and then they go and do the lab with the hands-on thing. So they're a little bit familiar with the nomenclature and directionality in the brain.
Yeah, you could hit that. You're right there, yeah.
I think there's a chance that I did not get right smack down in the middle because my insights were slightly different.
That's
Yes, it
fine. That's
It was
no
clicking
problem at
yesterday.
all.
I don't know
It's
that.
going to be even better. So we can see
Yeah.
certain things in the center one, and there's slightly different views from the next side around. So, but to go back to your question, I think either can work, because to make it an exploration, they know nothing about it, and just to walk them through it might provide a better sense of you know, curiosity and just that they are doing the discovering online. That the way that I described happens to be how my lectures get organized. And the more time, I mean, the students really love the labs associated with this, with the brain and behavior class. And so if I had the power, I would shift more of the time into lab explorations and less of the time into the lectures. So either way will work, I think. Okay, so let's take a look at what these things, we'll go and we'll do each brain individually, and actually maybe I'll do it once, and you guys look at the try and identify the same things. So this is a beautiful specimen because look at that. So see this white band here, and then there's this it goes down. This is the corpus callosum. Corpus callosum is the fiber pathway that links all the cells from this side of the brain to go to that side of the brain. So there's great interaction. It looks even bigger in the human brain. I can't cut this open, but it's there, it's prominent, and it's floating on top. But then you can see this offshoot that kind of diving down here. That is the fornix. The fornix are the output axons of the hippocampus. And the hippocampus is right below this, sitting at the top. Remember I said it was a C? The top part is here, and all those fibers are coalescing together, and they dive down, right down there on both sides. Okay, so those are really prominent fiber pathways to understand. Now, let's open this up. Look at that hole. You see that hole? There's a space. There's a huge space in there. What do you think that space is?
The lateral ventricle.
Lateral ventricle. So I always thought, I remember when I first learned this, I thought it was kind of cool that there's kind of a fountain in the middle of your brain. What flows inside the lateral ventricles? Cerebral spinal fluid, exactly. So that's the fountain in the middle. And in fact, the choroid plexus, which is the structure, this is probably choroid plexus, it's black and kind of gooey looking, creates the CSF. So it's being created and synthesized in the lateral ventricles, and it goes down from the lateral to the third to the fourth, and it goes out into the outer covering of the brain. And so I brought this to show you what, this is the dura mater, which is a little cap that goes all the way around. And cerebral spinal fluid is flowing in this space between the dura mater and the brain. And it provides protection for the brain, because what's just outside here?
The skull.
The skull. The skull is hard. I don't want my precious brain banging up on the skull
Do you want me to take a picture?
without protection. So it's like shrink wrap, right? It's all the way around the brain. It goes all up and down the spinal cord, and it is like I don't know if you guys know what Tyvek is. We sometimes
Stuff you used to cover like test tubes and stuff,
Yes,
right?
yeah,
It's
yeah.
like the thick shrink wrap.
yeah, exactly. But it's really, I mean, you could feel it. Look how
Oh, you mean like
hard-
parafilm?
much stronger than that.
Oh,
I mean,
yeah.
it's like,
But yeah, that's
don't
what
tear
I-
Oh
it.
yeah.
But
Oh. You know
let's
what?
see how far.
can
It's gonna be hard
torn.
because you know what? This is a tough mother.
That's right. Ha ha ha. So that joke
Bad
is
joke.
because
Okay.
dura mater means tough mother. But there's 3 layers of the meninges. Does anybody know what the second and third is? Dura mater,
Arachnoid mother.
arachnoid mater. What does that mean? Spider mother.
Spider mother.
And
Pia mother.
pia mater. What does pia mean?
Gentle.
Soft. Yeah, exactly. So, so all that CSF that gets created in the lateral ventricles goes down. It exits through these particular holes, and that's how it gets in this area right here. And so this protection, it's kind of like a waterbed for the brain. And the way that I like to illustrate it in class, that I think might work really well for you guys, is to ask the students, what food stuff that you can buy in the supermarket does this remind you of? It's a mystery.
The dura mater.
The dura mater and the protection that you get from the dura mater.
Sausage.
Sausage? No,
The
there's
covering
not.
of sausage.
But there's no space. There's no water in the sausage.
Yeah, I was just thinking like hot dogs or something where they have them.
Shrink wrapped? Yeah, but still no water.
It's a food.
It's a food.
The coconut, it's not really.
Close. It's kind of reversed from
Yeah.
the coconut. So I asked this question and I have this.
Egg.
No.
Okay.
Okay, I'm going to tell you. So I asked this question, and there's this mystery thing under thing, and I reveal it, and it's a box of tofu. Tofu, because hard tofu is about the consistency of the brain, and then the water that comes in it,
Oh, the CSF.
that floats in, is like the CSF, and then the plastic box is like the dura mater. And so I do a little demo, and I throw the tofu box at somebody, and they catch it, and then I throw it on the ground, and then I pick it up and I open it, and if I haven't thrown it too violently, the tofu is saved. And so I always make the joke that the packager of tofu must have been a neuroanatomist because he took the strategy that the brain uses. I mean, what's more precious to protect, right? The brain, and I don't want it banging up on the surface. So I as God-like person that I am, have designed this protection, this waterbed protection mechanism.
And it goes between the hemispheres as well.
It goes
looks
between the hemispheres. Yeah, exactly.
So it lets us go on roller coasters.
Yeah. This lump right at the front. So this is the front of the brain, right? Can you identify that lump in your preparation? There's a big lump there. It's a structure.
Is that this one?
Yes. Ooh, you got a big lump.
Right here.
Yes. Yeah. Oh, that. Yeah.
Big
Yeah.
lump
Yeah.
too.
Sometimes, yeah. Sometimes you have to dissect it out to find the lump. Like this one, you have this thin covering here that has to open up.
Because it looks like I got all my lump on
And
one
then-
side.
Yes. Yes. So that lump is called the caudate nucleus. The caudate nucleus. It's part of the striatum, and the caudate nucleus is important for motor functions. Part of the striatum is damaged in Parkinson's disease. The striatum uses dopamine, and the part that's damaged is, of course, the dopamine cells that die and cause the tremor and the motor control issues that you have. So there's lots of brain areas that are helping us control motor function. We talked about the cerebellum. Primary motor cortex is right up here. That's obviously critical. You have damage to this area and you're not able to move your body voluntarily anymore. But the other structure that's important for fine motor movement is the striatum, and we're looking at part of the striatum called the caudate. So it forms the lateral wall of the lateral ventricle. And now we're going to move down here. Can everybody see a circular kind of structure
Would that
right
be
here?
that little ball?
This ball?
Oh,
Yes,
okay.
that ball.
That was that
Right
one.
there.
Yeah, there's several balls. Sorry. Try and identify the correct ball. That is the ball of the thalamus.
Right.
Okay, right and left, and then there's a little ball up here, kind of hanging off the back end
Yeah.
of the thalamus. Anybody know what that is? There's the thalamic ball, and then there's this littler ball.
That's the
pituitary?
pineal gland.
Pineal gland. Ooh, very good. So why is the pineal gland interesting? Ew.
It secretes melatonin.
Yes.
Involved in our circadian rhythm, sleep-wake cycle.
That's more the suprachiasmatic nucleus. I just got brain juice on my keys. Between those later. Pineal gland, seed of the soul. It was called the seed of the soul. Why do you think it was called the seed of the soul? There's something unique about it.
Does it produce hormones that make you dream?
No, I mean it produces hormones, but that's not the thing that made people think that. Thank you.
It's not
That
related
is
to.
the seed. Oh, these are the bad ones. Oh.
So is that
only-
related to melatonin?
Yes.
Wendy, do you have paper towels?
No, I'm thank you. There's only one of them. Pineal gland. It's the only structure that is singular in the whole brain. And so they thought, oh my God, look, everything has a double except for this, so it must be the seed of the soul. Of course, that's not true, but the point is you even though it kind of looks double here, if you
If you didn't dissect
If you
straight
didn't
through
cut-
the middle, you
exactly.
get a whole little
Yeah,
nugget
one.
in
Yeah. Oh,
there.
you exactly. Oh, yeah.
We got a
See,
good
there, there it is. Look how big it is. It's huge. It's relatively big in the sheep brain. So,
And is that centered, or does it skew to one side?
it's centered. Yeah.
My knife just skewed. Fortuitously.
Yeah, exactly.
Okay. Next, we're going back to the next bump down. So we have the thalamic bump, kind of towards the front. We have bump of the pineal gland, and then here's yet another bump. And the best way to look at that is to put it back together and do this. Whoop. Can you see those? Mm-hmm. There's like 4 bumps.
Oh, yeah.
If you look at it in the back, the bump on the top is big and the bump on the bottom is little. Can you see that? So that is the midbrain, the superior and inferior colliculi. Yeah. And so what are those? What are the functions of that? Those structures, inferior versus superior colliculus.
I think they're involved in like automatic responses to sudden noise and sudden visual stimuli, like flashing lights.
It has something to do with vision, definitely. The superior colliculus is important for visual processing. And remember I told you how good the sheep is about vision? Which one is bigger, superior or inferior colliculus here?
Superior.
Superior is huge, it's humongous, because the sheep has a very highly developed visual system. I was telling people that sheep can identify and discriminate all of the other sheep in their flock and discriminate them from each other and from other sheep not in their flock. And that's hard because all sheep look identical. Sorry, that was just a joke. And inferior colliculus, the smaller one, has to do with auditory processing. So you probably won't get into that in your groups, but if you follow the auditory pathway from sound going into your ear, going up to primary auditory cortex, it passes through the inferior colliculus as one of its processing pathways. But it's really, in fact, you might want to take pictures of this colliculus side view and then kind of have somebody pull it open so you can see what it looks like in here. Okay, so why don't you guys do that so we don't get too far between the, and if you want me to hold something, let me know so you can take the picture.
I'll take you up on your office.
Next, I want to do a little bit of a dissection. First, some coronal sections, and then I want to dissect out the hippocampus as the pièce de résistance. Okay, so let's do. I'll do your brain.
Okay.
So just keep it there. Use the knife. And so I just want to start to cut through the brain. And the cool thing here is that you could really see how invaginated the cortex is, even more from just looking at it at the front. So just trying to, and this is all of the prefrontal cortex in the sheep. And you can see it. Okay. And what I'm looking for is the start of the lateral ventricles. Oh, there they are. That little hole there, which is there, is the start of the lateral ventricles. Can you see that? So, and then what is this big ball here that's making up the lateral wall of the lateral ventricle? See that wall? Round thing.
Is that the cerebral peduncles?
No, peduncles are way down here.
Oh.
We're way in the front.
I guess it's part of the seahorse.
Oh, close. That's
The
back
amygdala.
here.
The amygdala.
No. Amygdala is back here. Here's the temporal lobe. We're still even in front of that. So take it apart and look at that first lump.
Let's see.
It's cutting through the first lump that we saw. See that? What was that lump?
Oh, that was what did you call it?
That lump. The caudate.
Caudate nucleus.
The caudate nucleus. So you can really see it beautifully. This round structure here. That's the caudate nucleus, and then you can see it's forming the lateral wall of the lateral ventricle. So cut until you can about see that that level. Okay, so the hippocampus is a C-shaped structure that goes right about here below the cortex and goes down into the front of the temporal lobe. And sitting right in front of the hippocampus is, what other structure? What's right in front of the hippocampus?
The amygdala.
The amygdala, yes.
I thought the amygdala was behind it.
No, amygdala is in front of the ventral part of the hippocampus. So what I'm going to do is take one of the hemispheres, and so remember where that fornix was? There's the fornix. That means the hippocampus is right there. All the fornix is coming out of the hippocampus. You just can't see it as well. So I'm going to use this as a guide, and I'm going to cut down. Also, you can appreciate all the invagination. And oh, see that? I hit it. And so that's the top of the C. There's a C that's going to, and the what we're looking at right there, so we see that is the top of the C. So I'm going to follow the C all the way down. And so to do that, let's see. Sorry, I'm going to cut off the cerebellum. You're in my way. Oh, see? It's getting bigger, top of the C. And note that the ventricle is surrounding the hippocampus. So we go down.
You can really see it's a separate structure.
Yep.
You just like a piece of garlic.
It does, yeah.
I will never look at garlic again, thank you,
Most of
Daryl.
those sections look
See?
like food, you know.
Look, you can see it go all the way down.
Oh, yeah.
And that black stuff there, that is part of the choroid plexus. There, that's the whole C, from there to there. And you can see this is turning into the fornix. It goes down like that. So I could take more off right here. So the amygdala would be right here. It's hard to see the amygdala. The amygdala doesn't have the same kind of structural integrity that the hippocampus does. But you can peel the garlic clove even more and take it out. There. And also what you can see is the hippocampus is basically making it's like spooning the thalamus. There's the thalamus. So the top of it doesn't exactly spoon the thalamus, it kind of goes out to the side. But it's sitting right on top of this ball, which is the thalamus. Okay? So why don't you guys try, you can do the other hemisphere. And
So
do
we're
that one.
gonna start cutting from the outside.